Well, today it's my pleasure to talk with Franz Vollenweider. He's a professor at the University of Zurich, where he's co-director for the Center of Psychiatric Research, the director of neuropsychopharmacology, a big word, and brain imaging. And he's the director of what I understand to be a Center for Consciousness Studies that he started. Is that correct?
I started it in 94.
Yeah, in 94.
Yeah.
And before we get into the topic of psychedelics, could you, so you're in Switzerland. What's your background? Did you grow up in Switzerland, and did you have an interest in science as a child?
It started very early. Yes, in fact, I was very attracted by biology when I was an adult, a young adult, maybe not adult, but a kid already, 12, 14, 15. I was collecting animals and all these things and got really attracted by nature. And I had an uncle who was a quite famous environmentalist. A lake researcher, and so we had a lot of discussions about the environment and all these things, and that attracted me to go into that somehow, but I didn't know how at that time, because the other kind of other ego in me was more an artist. I was always playing drums in a band and had different bands, so music was an alternative to go for. And so later then, maybe at the high school, I came across, had a very good teacher in German about Nietzsche and Freud and all these things, philosophy of the mind. Oh, well, what's that? I thought. And I was looking around a little bit and found some books about the brain, but they went back 100 years. Very funny. And so I thought, okay, that could be something. So the other thing was then I was really attracted by the question, what is the self? What is the ego? Why do we have a center? And how is that implemented in a brain or in nature? And that was the starting point, this question of the self. I have a self, and apparently we have an unconscious and the consciousness, or what I learned at that time. And I thought, oh, wow, this is interesting, the dreaming of these things. And then, yes, I thought to bring together my interests in natural sciences and these more psychological questions.
And
that
so
was
then,
the starting point. When
you started college, then you were at the beginning planning on going to medical school and psychiatry?
No, not at all.
No.
Driven more by biology and these environmental questions, I started with biochemistry. So that was a new discipline at that time in Zurich, where people had a lot of interdisciplinary research, biology, chemistry, system biology. And I started with that, system biology and biochemistry. And then I found out, oh, well, this goes down to all little bits of a molecule. Maybe I have to enlarge that a little bit. So yeah, then I switched after 2 years into the medical department. So you have more the system, less just the chemistry of a cell. So I was more interested in becoming aware. I feel more attracted by the whole system and the interaction of individuals and peoples and animals and plants and these things.
Yeah, I think it's always better to have a foundation in basic biology and biochemistry, and then move up to the more complex questions of how all this is working together in the nerve cells and so on, which you've been doing now for quite a while. Now can you talk about the historical perspective on psychedelics? A lot of our viewers will be aware that LSD was first synthesized by Albert Hofmann over in Switzerland. And then, of course, in the United States, there's this big issue in the 1960s with a lot of kids were taking teenagers were taking psychedelics, and for the most part, they seemed not to terribly affect them adversely, you know, but there were these odd reports of someone having like a psychosis, almost like a schizophrenia-like condition. But anyway, can you give like a historical perspective?
Yes, I can do that well. When I get attracted in psychedelics, that was in the second year, where I learned about molecules in plants and mushrooms and this stuff. And I realized, oh, this is exactly the interface I'm interested in. Molecules affect your psyche, your brain, can alter your vision, etc., etc. And then I found out that Dietrich was doing some research at the University of Zurich. And Dietrich was, let's say, on the path that started in the 40s, 50s in the same institute at the University Hospital of Psychiatry in Zurich, where they did the first study into LSD in 1984. Yeah. And Dietrich was still around, and I called him up and I'd asked, oh, why? This is heavy work you're doing. And he said, yes, since 10 years, he was investigated what is the common core effect of different inducers of altered state. And then I thought, oh, wow, this is great. And so I learned then through him and other people there at the institute, which I frequently visited beside my studies. Oh, they are doing something. And then I called up Albert Hoffman, who was still alive, lively person, and invited me to come down to Basel and talk with him my interests. And he said, you're on the right path. Medicine is better than biochemistry. So you have the access to the drugs. So, okay, it was a good decision. And so the first study they did at that institute, where I'm still am now, was in 84, as I said, into depression. So Contra did a study in depressed patients. They also did a lot of studies in healthy volunteers at that time in the 50s, and even in schizophrenia. But you may have heard that the institute was the son of the famous Bleuler who coined the term schizophrenia. And they were interested, is there something going on in the brain, a molecule or an endogenous molecule that may induce this schizophrenia-like symptoms? Is that comparable with psychedelic experiences? And that was, for me, the right question. What's similar, what's different? But I was still a student. So then at that time, it was about 85, 90, I went through the university. I was finished at 84. And so I was looking around, and Dietrich quit his job. He got retired. And I went then to an institute of brain research institute to do really some basic stuff in animals, in the hippocampus, which is important for learning. So, but it never went out of my mind, this more system-like things, questions of a psychedelic affecting the whole psyche, the environment, and the interaction with people. So then I saw that there were some studies going on in Switzerland, some group of psychiatrists, the so-called Society for Psycholytic Therapy. They were heavily engaging in treating people with LSD and MDMA. So I got connected with this group, which was connected with a larger group, with the European group of psychiatrists that were interested in psychedelics. This was the ECBS, founded by Albert Hoffman, Dietrich from Zurich, and Leuner from Germany. So we had three big guys during the 80s up to the 90s. And so there was a tradition, but that people looked at this stuff still in Switzerland. It was never completely blocked. So, but then I formulated a real hypothesis that I wanted to test with Dietrich, who used hypnosis to induce altered states, drugs like mescaline, psilocybin, MDMA, cannabinoids. He used meditation, deep meditation, over how you say, stimulation with white noise, and was looking at this common core of the effect. And I thought, okay, let's do something in parallel, and designed a study using psilocybin, ketamine, and amphetamine. These three drugs interact, as we had known at that time, with different neurotransmitter system, and may end up in a overlapping effect, in the brain. So we hadn't any idea about that in '92, where I started with that study. It was the first brain imaging study I've done, comparing different pharmacological inducers to tease out some brain regions that may be responsible for the effects we see in these different drugs. And then we started to correlate this brain network findings with psychological findings from Dietrich's rating scale. So that was the really start in '92 into, let's say, the biological foundation or the biological mechanism of possibly underlying the psychedelic experience. So that was the way back. There were a few groups in Europe, in Germany, that still have done a little bit with mescaline, and does the neuroimaging 2 years before us. There was Evi Goussoulis in Germany studying psilocybin. But at that time, most of people were not running for the clinical effects, for instance, that these drugs have a beneficial effect on depression. They were looking at the basis. How are these symptoms generated? What may be altered in terms of biological mechanism that lead to these psychological experiences? Also, I myself started like this. I wasn't really into the clinic at that time.
Yeah, and now there's just an explosion of psychedelic research over on this side of the Atlantic, as you know.
Yes.
And where I'm at Johns Hopkins, they started a center.
Yes.
It's small, Roland Griffiths and going, but it looks very promising in treating depression. Psilocybin has been mostly used and for reasons that are, I don't know if there's really a scientific reason to use psilocybin versus LSD, but somehow because of what happened in the 1960s here, the investigators felt there'd be less pushback.
Yes.
In studying, the mushroom chemical.
Mm.
Now, you talked about these different psychoactive chemicals found in plants, mushrooms, and then ketamine, which actually was an anesthetic. They
Yes.
act on different neurotransmitter systems. Can you just kind of briefly describe the core neuronal circuitry throughout the brain? Because a lot of people, they've heard of dopamine, they've heard of serotonin, but it turns out glutamate is the most prominent neurotransmitter. All the excitatory circuits use the amino acid glutamate, and these other neurotransmitters affect behavior, and probably these drugs, right, by modulating the activity of those glutamatergic neurons, the neurons that use glutamate.
Yes, that's correct. What is important that we know now that the so-called classic psychedelics like psilocybin, LSD, and mescaline, and DMT from ayahuasca brew activate a specific serotonin receptor. They have a high affinity to the serotonin, to a receptor family, and are partial agonists. They activate this receptor. And in contrast to this, other drugs like ketamine blocks particularly a specific glutamate receptor, so-called NMDA receptor, which is important in learning. And these are two different things, and how are they linked together? So in about 2000, George Akajanian from the Yale Institute made a discovery with other people that this classical psychedelic that activated 2A receptor and downstream somehow increase glutamate concentration in the brain. That was the starting point that we followed. And also in brain, with brain imaging, we saw that many regions in the brain acutely get activated, and much more than possibly just through 2A activation, but maybe due to glutamate research that it comes up. And that became more and more important, that downstream effect on the glutamate system. And surprisingly, we have seen that also with ketamine in our first brain scans, that ketamine activates very similar brain regions in the frontal cortex and in the frontal parts of our brain. And that was a question mark, because if we went back to schizophrenia patients, they had a reduced activity in frontal areas. So, but in this acute, highly lively, full of experiences, psychedelic experience, some important regions lighted very strongly up.
You know, Franz, that's really interesting, finding that the ketamine increases activity in some of the same circuits as, say, LSD or psilocybin. And now, one transmitter we didn't mention was GABA, which is an inhibitory neurotransmitter.
Yes, yes.
And it turns out that many of these so-called interneurons that use GABA have very high concentrations of this particular type of glutamate receptor, the NMDA receptor.
Yes.
So if you inhibit an inhibitory neuron, then that will increase the excitability
exactly
of the neurons those normally inhibit. Do you think that might be what's going on?
That's certainly a part. There are lots of GABAergic interneurons between the major players. These are the, let's say in the cortex, the pyramidal cells. They use glutamate to get excited. And the interneurons are like a brake, they're in balance. So there's a balance between excitation and inhibition. So, and yeah, GABA is very important. And we have done two things. On the one hand, you can activate the GABA receptor, then you bring down the overall effect of psychedelics, but you cannot blur the visions you have. But if you do a 2A antagonist, which we have shown almost 25 years ago, then you can completely block the effect of classical psychedelics.
With what? Sorry, with what?
It's a serotonin 2A receptor antagonist.
Oh,
for instance.
that's
ketanserin.
Ketanserin, yeah.
Yeah, blocks completely. Pre-treatment blocks the LSD effect, the psilocybin effect, and almost all of the ayahuasca effect. There needs more studies to be done. But with ketamine, not. Ketamine we couldn't block with the GABAergic, with the 2A antagonist.
Yeah.
We could modulate it with the GABAergic agonist. And this speaks that somehow the glutamate that comes after the serotonin system is a major player. And it is anyway interesting if you block the glutamate system, these NMDA receptors with ketamine, that glutamate goes up. So it must be a circuitry. So like a cascade of interacting systems that in the end all enhance glutamate. So it's not so easy, but they converge in like a crossroads. And
we've done. I did a lot of work in my lab over many years on excitotoxicity.
Mm-hmm.
You know, because neurons can actually be killed if they're excited too much by glutamate.
Yes.
And it turns out, this was a long time ago, a guy named John Olney at Washington University. He showed if he gives a drug that's similar to ketamine, it's called MK801, to rats, it will actually cause
Localization.
excitotoxic, yeah, because it's affecting the GABA.
Yeah.
You know.
Yeah.
Yeah.
Yeah. As I remember when I started, when I was still in brain research, I worked in the hippocampus and used MK801 instead of ketamine or PCP at that time.
PCP, yeah.
And we exactly saw that we have this rush of glutamate, but also this localization of anterior cingulate. So that means too much glutamate is not good.
Yeah, yeah. Now, go ahead and talk about functional magnetic resonance imaging, how you've done many, many studies looking at how these drugs, the psychedelics, amphetamines, so on, affect neural network activity. What is functional MRI and why is it so valuable in understanding the effects of these drugs on the psychedelic experience, for example?
Also, fMRI or functional magnetic resonance imaging is a neuroimaging tool that allows us to measure indirectly neural activity through measuring the Ball signal. That's saturation is oxygen, a signal that gives us a hint what the neurons are doing, whether they get excited or they're less active. It's not that we directly measure it. So it's also a little bit difficult sometimes if drugs affect the blood flow. Then we have to take a lot of noise. Basic global changes we have to partial out through mathematical approaches to see really how these regions get activated or deactivated. And it needs a lot of tricks, but it's the only relatively fast method that gives us a good information in a three-dimensional space in the whole brain, deeply down. And we use that to look at the effect of psilocybin, LSD, MDMA, all kinds of drugs in resting states. There are many different approaches to using this technology. One is we look at people, they are at rest, eyes open, eyes closed, looking on a red cross in the scanner where we project on a little monitor. That's the so-called resting state condition. They do nothing. They should not think, but certainly they think and have emotions and so on. But we call it over time as resting state, let's say 10 minutes. We measure that episode, sometimes 15 minutes, and get a global information about the whole brain, how regions interact. That is mathematically teased out of the data. You see activation, deactivation signals. But from this, you can look what brain areas go in sync, which are synchronized. They go with the same wave, let's say, they are coupled. Yeah. And so this is a measure people use. It's called connectivity, how they are connected. Drugs can increase connectivity or decrease, reduce the interaction of brain areas. So you get these famous maps where you see quasi the interaction, the interplay of all these regions. And if you use then certain algorithm, you can tease out basic, we call that intrinsic networks. These networks are responsible for different things. For instance, one people speak about is the default network. This is a network when you, let's say you have your eyes closed or open, you're silent, you think about yourself. Yeah. And this network has several nodes very symmetrically through the midline structures of the brain, our cortical areas, and they give you a sense of yourself. That was a famous discovery maybe 20 years ago, I can't remember. And everybody was speaking about default networks. So we just think about ourselves, and that has to be revised. So we can't say daydreaming, you're in a daydreaming state. It's also important if you differentiate between yourself and something else, some other person. So the different nodes in the default network are a little bit specialized on on different tasks. So if I look at pictures of myself, the default network goes in part activated in frontal regions. But if I look at a photograph of other people, partners or whatsoever, famous people, more posterior regions of the default network are important for social interaction. So this is one. Other networks, intrinsic networks, are responsible execution, commanding, starting motor activation, executive systems. Very important is the attentional system, a salient system that gets activated if something new comes in, novelty detection. So these networks get quite modulated by psychedelics. Most people, yes, sorry.
The word salience is the
Salience detection.
Your conscious impression of the importance or value of the information coming in.
Yes, exactly.
Yeah. Okay.
And this is a cortical network, and certainly if you speak then about values, then very deep structures in the brain gets co-activated. The dopamine systems, they're very important for modulating and adding emotion on what you perceive. You know, it's always a mix of many different systems, but so at least.
And then these systems, ultimately, one function of these systems interaction is for the person to make a decision about something.
Yes, for instance, in the executive network, that's decision making is that implemented also. Yes.
So with psilocybin or LSD, where do you see changes in the connections, the communication between different brain regions?
Yeah, but one is, let's say, a robust finding across different studies that the default network is reduced in its activity, and the interaction, let's say, with the salient network, who is responsible for novelty, this interaction is loosened, and interactions in the posterior cingulate towards other brain regions, as to the temporal cortex, are loosened, less strongly interconnected, so just functionally in the psychedelic states. And that allows, so that's the hypothesis, to have a new perspective on incoming information. It's not immediately so strongly, tightly processed, like you have a little window, like a veto. You can watch your thoughts, your emotions to be less involved, you know. And this reduction of the default network has been linked in a few studies with a reduction of self-centricity. So your ordinary self, which is, let's say, founded in the activity of the default network, gets reduced. So you become a little bit more open. You don't interact as fast. We are more in silence. And I would say you are decentered, like the Buddhists say. You are less in the stream. You have a certain distance between what's going on where you are. That allows you to have a new perspective on emotions and on cognition. And the reduction of this self has also been linked to be less driven by, let's say, ruminations, or you're in circles. You think about yourself in depression. I'm bad, I'm not, my self-worth is down. You always have this negative spiral, and you get a certain distance to that, and you're less driven because your self is reduced. That's the overview about these processes.
Now, what happens? What are the correlations with actual hallucinations? You know, visual hallucinations.
What we found, for our hallucinations, we also used three-dimensional brainwave detection with EEG, electroencephalogram, and nowadays you also can through mathematical approaches, you can have three-dimensional pictures, but it's much faster, because hallucinations come and go, and the time resolution with fMRI is 2 seconds, with EEG is milliseconds. So we did a lot using EEG to follow when people had button presses, and we used other paradigms to go for hallucinatory phenomena. We see modulation of the visual pathways, which are normally activated through external visual information that comes into the brain. These visual pathways gets activated at different streams. There are parts that detect, let's say, colors. There are other parts that detect movement of objects. And all these different parts who are detecting features of an object have to be integrated in the frontal cortex again. So it's like you have all the specialists who look for, oh, what's going on here? Oh, it's moving. Now let's give a signal. It's black and white. Oh no, it's color. And you have all these different neural networks who work on teasing out that external information. But it has to be integrated in the end. And what we see along this stream, that certain areas that are important for movement are enhanced in activity, so that things go faster, that certain pathways that are important for detecting the overall gestalt are less active, so that may blur the real physical form of an object. And along that stream, it's important whether the information is emotional or not. So you have also, you add emotion to what you see. And here it became very interesting. We found that in 2012, the first time, that if you show people friendly faces, happy faces, angry faces, which is very important for social interaction, we interact with seeing faces, that they are completely different modulated with psychedelics along this visual stream. Let's say angry faces and anxious inducing faces were reduced with psilocybin, but not the happy faces. The happy faces were normally detected and processed, but not the anxious. And then we thought, oh, this is interesting for psychotherapy. People have an external information, like good and bad information, but could that also be important for internal information from the memories? You have bad memories, you have good memories. And could that be an explanation that people are, what I use the word, more decentered to their emotions, that the negative emotions, they see it, but they're not so wild. They are not so, I would say, oh, I don't know the English word for that, bedrohend.
Profound, or yeah.
Yeah. And that was the starting point for us to go back to the clinical application. Because most of the visual experience is a distortion or is a recall of old images or memories. People report, oh, I experienced myself like being in my room when I was a child and see all these puppets and these things. So it's a hallucination, but it's also very personal. It's very self-relevant, and it's almost emotionally loaded, and but it's interesting that the visual system that normally is responsible and areas of the back of the brain who are important for storing this kind of information get activated, even with memories, recall of memories, and additional subtile illusions or hallucinations.
Are there any similarities between what happens in REM sleep, which is a dreaming state, and what's happening in the neural networks with the psychedelics? Or are they very different?
That's a good question. In one I say one feature or one property of the REM sleep is that people have more dreams, vivid dreams. And it's also linked to the temporal cortex along the side of the brain. And there is some evidence that psychedelics, this is also involved in hallucinations, temporal cortex in our data, is important for the REM sleep, hallucinations or dreaming phases. It might be an overlap, but there is no real data at the moment demonstrating that. The only thing what we have is 3 very old studies from the 60s, where they gave people low dose of LSD or psilocybin here in Switzerland and observed an increase in REM phases. But that are very old data. So
there seems, just based on my own experience with dreams, there's kind of a evolving story going on. Is it with the psychedelics? My impression is it's a little more random. It's not an evolving story. Is that true?
Yeah, it's maybe more a mixture. It's maybe more, sometimes more overwhelming. I think this depends strongly on the dose. We have done a number of dose response studies, starting with 5, 10, 15, 20 milligrams of psilocybin up to 30. And then you see that the doses, let's say up to 20 milligrams or 18, I always say a strange number, 80 milligrams, there's something going on. There's like a border. When you go over that, it becomes vivid and vivid and vivid. But below, it's more dreamlike. I'm convinced, because with 5 and 10, we have more illusions. With 15 to 20, really overlaid hallucinations, or things that have no external equivalent pop up, starting with geometric patterns, with seeing flowers, with seeing little animals coming out of a vase, or whatever. This is what we call a hallucination. But there's a lot at lower dose, a lot of illusions. Just the geometry of the space is distorted or changed, or maybe very nice, but and that is certainly, to my understanding, more dreamlike. And what's interesting, people have really clear insight. We train people their attentional capacity. We use a mindfulness technique in healthy, but also in patients. When we treat depressed patients, we learn them to focus inward and to keep attention. And then it's much more story-like, But if you just follow everything you see in the outer world on a first time on such an experience, there's so much going on that's funny, interesting, wild, or anxious, inducing,
So in those studies, typically people think of someone, while they're having a psychedelic experience, to have a good experience, you'd be like in a quiet place, kind of a safe environment, not any input coming in. But you're saying you're doing studies where you interact with the person while they're...
Yes, mostly we interact with the persons doing a certain task. Let's say when we were interested, how do they process emotional phases? We have to flash them with pictures. Or if we look at social interaction, people play a virtual game versus somebody else, how they interact with the avatar or with a real person. We did also with a real person in the scanner or off scanner with EEG. You can do that easily. That's very different from resting state activity. The question here is, how does a certain task, where you have to do something, you have to process something, how does then the drug modulate that capacity you normally have? It's quite a different thing.
Okay, so you talked about the 5-HT2A serotonin receptor, and that's clearly established as being the target or the receptor affected by the psychedelics. And my understanding, this receptor generally, it enhances the excitability of glutamatergic neurons. Is that correct?
Yes, in the deep layer of the cortex, some feedback system axons go on other pyramidal cells and then they get activated. That's the theory, we think.
And I was reading, and I briefly mentioned this when I talked to you, briefly in preparation for this, that if people who've looked at the levels of this receptor, the amount of the receptors on different neurons in different brain regions, find that in the prefrontal cortex, there are certain pyramidal neurons, I think in the deep layers that you mentioned, that have much higher amounts of this serotonin receptor than others. Could you talk a little bit about that?
Yes, that's true that in the frontal cortex is the highest density of 2A receptors, and it gradually goes down to the posterior part of the head cortex. But the differences in the density is not that tremendous. There's much less in deeper structures like so-called basal ganglia, limbic system. Thalamus has not much. We think is only one study that has looked at the occupancy of the receptors. We have done one so-called PET study with positron emission tomography, where we have labeled a 2A agonist and then looked how is it displaced by psilocybin. And we found, in fact, that if people are in an oceanic state, we call it oceanic boundlessness, blissful, no, let's say, vanishing of time, not being completely aware that you're in space, you're really in yourself, happy, blissful, peaceful, that this state, when we map that state in terms of 2A receptor occupancy, we found high occupancy in frontal areas, frontal middle cortex, temporal cortex, left insula, and mostly frontal. That's to say. Now another much more recent study using a little bit better traces and neurotechnology found the same, that with the occupancy, with the degree of occupancy of serotonin receptors, the symptoms go in parallel, or the intensity of experience goes in parallel, not the symptoms. It's too pathological.
I was so with the psychedelics, it's mostly visual hallucinations, not auditory. Is that correct?
Auditory, yeah, it depends. Auditory is quite modulated. Most fancy story was when we played music in the beginning of our studies to people with a very small, tiny stereo player. And they said, wonderful, oh, this is the best sound I ever heard.
Oh.
It was rubbish. Because at that time we hadn't all these nice tools we have today. Now, often it's also an experience that people have the idea that they go with the sound. The sound is a driver if it's not too much music, you know. We have done a study where we have looked how do people process some of their self-relevant music pieces. They had to bring 10 MP3 files with very important music to them. Not good music, you know, but important. But they've been fallen in love, or they have changed I don't know, way of living. And then our physicist, we worked together with Jonata in Los Angeles. They are looking how music modulates emotions, you know. And they mixed this sound and made use the same physical stimuli. They changed the sound pattern, but they used the sound snippled, with snips and put it upside down. And then we used as a neutral sound, which was similar in terms of rhythm and melody, but something completely they hadn't known. And surprisingly, their own sound was a little bit more emotionally, a little bit more self-relevant on LSD. But the rubbish was completely novelty. They found it fantastic. And has led to activation of brain areas that are important for, as we said, value, emotions. This was really interesting. That's something you heard first time under this drug, which you haven't known, gets you you get so alert on that. I didn't
No,
believe it.
and people with schizophrenia, they commonly have these auditory hallucinations where
Yes.
they hear a voice that isn't
Yes.
there.
Yeah.
Does that ever happen with?
Not much. Some people had told us that they had a kind of voice-like experiences or strong thoughts, you know, they were their own thoughts that were overwhelming. And sometimes a few people were not sure, was that real? Is there something supernatural talking to me? And they got afraid of that. So, but it's rare. It's more modulating the sound.
Now that gets me thinking about the language. So animals like rats in the laboratory,
Yeah.
these psychedelics have behavioral effects on them. And they obviously don't use language, at least the way we do. So I'm wondering if that, like, you know, this is like an ancient thing, and the language hasn't been incorporated into these psychedelic experiences. Does that?
You mean that language is
Well,
evolutionary, a new
you say
thing?
you don't hear voices, right? That's language.
Yeah.
And people probably don't see words. It's always like visual things that are, I guess, not related to language at all. Is that true?
Yeah. I haven't thought about that in details, but makes sense because the vision system is the largest system in our cortex. Auditory is very small. It's evolutionary new, but what they use within the visual system is a lot of symbolic experiences, you know. Maybe language is a more higher structured symbolic information.
Yeah.
Reduced to an alphabet or 0101,
Yeah.
whatever language you speak.
Well, what about numbers? What about numbers? Math? No.
Numbers, not so much. What's interesting, sometimes people are a little bit, you know, expectation plays an important role with psychedelics. We had some chemists that were always experiencing molecules, how they attach and react, and so they had all the visual stuff overloaded with their job somehow. And one said, I feel like to being in a blood cell and I see all the metabolic pathways.
Oh.
And I said, have you learned that? And he said, yeah, certainly once, but I experienced that now. It was funny. So it depends a lot of possibly what you bring with you, you know, and we haven't talked about that, but expectation plays an important role in terms of having a good or bad trip and all these intentions people do.
Could you talk about the therapeutic application, you know, clinical applications? Depression is one that looks very, very promising. Mm-hmm. You mentioned addiction and some of the work done over there in the when was it, 50s, 60s? With addiction, alcohol addiction?
Yeah, it was in the 60s already. They did some pioneering study in alcohol addiction, yes.
Are there do you know any trials going on now?
Yes, this is a large trial at the NYU in New York by Michael Bogenschutz. He has looked, now he's showing the results recently on the patients, and it looks quite promising. And they measure the amount of drinking after they get treated with psilocybin. Some are detoxed in the beginning, then they measure after the psychedelic experience how much alcohol they still drink, and that drinking rate went down significantly, more than just placebo, certainly. It was a well-designed study. That's one important study. There are smaller studies. I don't know whether Johns Hopkins is also doing, but the most we do one in Zurich. We also look at the relapse rate with psilocybin. People get a 2-week preparation phase where they are completely off alcohol. Then they get some mental training, and then they get the psilocybin. And we're still going on. We combine it with brain imaging. The New York study did not use brain imaging, but we wanted to understand the mechanism better. As alcohol addiction is certainly a thing. Some people have thought about cocaine addiction, but I'm not aware that something really is going on in that field. Most studies at the moment look at therapy-resistant depression, about 5 ongoing studies worldwide, and about 6 just in depression, so not therapy-resistant. But they should be finished in a year. But there are a number of studies that have been published, some pioneering studies, some open studies. That means people were not blinded, really. But 2 blinded studies, including a new one from our lab, have been completed. We have completed a placebo-controlled study. That's a hard thing to do. It's discussed. You take placebo, you take another drug, but in terms of regulation, making a point, once you have to do a placebo study, therefore we decided for the placebo.
Yeah, well, the patient will know whether or not they got the drug or placebo.
There were a few, yes, exactly. And then you have to prepare the patients, and that in the beginning, they can express their disappointment, not just on the trip. So they're less disturbed than when they get the placebo. But we were surprised that of 25, 4 had a strong psychedelic experience. So we said, what the hell? Is that still expectation? Now we're looking at the brain responses in detail, whether they do the same like the psilocybin. They were exactly in their experiences telling us this and that happened. We saw that also with healthy volunteers over the many years, that a number of people just react. You give them something, and you may heard if you give depressed patient a placebo, also have a response rate 30 to 40%. So one of our mentors always said, you go better to the cafeteria with the patients and give him the placebo. That helps us. So expectation is a real problem to control, and the blinding. But yes, most of these studies found a reduction within a day, and then at least in 2 weeks, 3 weeks, with 1 or 2 doses of psilocybin, first mostly small, 10 milligrams and 25 in the US. Most studies also in England, they are made like that. And show enduring reductions up to 5 weeks, sometimes 3 months of depression. Yeah. So only one study, the one from Robin Carter-Harris in London, had published imaging data so far. And they suggest that networks work better together after the psilocybin treatment, the global interaction. We can't say about our subjects, how they do, because we're still analyzing the data. But yeah, we don't have a lot of imaging data so far.
A remarkable feature of the psychedelics with regard to the depression is, 1 or 2 exposures give long-lasting relief, which contrasts with the currently prescribed drugs for depression, which people have to take every day. Now, there are some evidence from animal studies that there's actually structural changes that occur in the neural networks. Maybe increased number of synapses or strength of synapses in certain regions. And so probably this goes back to what happens with learning and memory. You mentioned you initially worked on the hippocampus, which is where learning and memory is, for really a number of important reasons, it's been studied. And there, when an animal learns and remembers something, there's actually increase in size of synapses, or even over time, increase in number of synapses. So it's hard to look at this in humans, but there are some data from animals that suggest
Exactly.
it. And those long-term changes require changes in gene expression. Certain genes are turned on that encode proteins that are probably involved in this. In the case of depression, one such protein, BDNF, brain-derived neurotrophic factor, has been intensively studied and seems to be very important in the antidepressant effects of electroconvulsive shock and even of the serotonin norepinephrine reuptake inhibitors. And so, yeah, I think these studies, like you mentioned, this other groups looking, everything you mentioned you've done has been looking at kind of the acute or soon after taking the psychedelics, what's happening. But
Yes.
there's been much less done, waiting a month and then see anything.
Yeah, exactly. That's the reason in our study with depressed patients that we did a follow-up study after 2 weeks, 3 months, 6 weeks, 3 months, to study brain networks alterations. And we have 2 hypotheses based on many studies we have done in healthy subjects. Insofar, you can just look data-driven. You can give a psychedelic, then use all of sophisticated tools to analyze brain imaging data. But we used a hypothesis-driven approach. We said, okay, let's look at, is there a change in brain networks that are responsible for this self-centricity or the egocentricity, that people are so focused on themselves? Is there really something going on in these networks over time? And the other most important for me is there a change in deeper networks, like including the limbic system, who is responsible for emotion processing, adding labels to emotions, and for anxiety, happiness? Is there something on the long run change? So that's we call simply emotion processing areas or networks, and that's important that you have a certain hypothesis.
And as the amygdala,
it's
a relatively small structure compared to other brain regions. Can you get a good signal on fMRI?
Yes, amygdala can still get a very good signal. And we have seen, as you said, acutely psilocybin brings the amygdala activity down with the interaction of other regions in the basal ganglia. And Fred Barrett from Johns Hopkins has shown then that this reduction of response to anxiety-inducing stimuli is reduced over 1 month in healthy subjects. So the network changes endure at least 1 month. And now we are very curious, is that the same in depressed patients? Because they were less fearful to fearful stimuli. We don't know at the moment. That I can say to this. Other people say that the default network or hubs, also nodes with the default network, are less close connected in terms of functionally connected, that they get relaxed, and that goes along with alterations in self-experience. But it's still all on, how I say, work in progress.
I'm aware of some studies providing evidence that the size of the hippocampus in depression may get smaller, and then in people that recover, it may get bigger again. And the thinking there was that, the dendrites, which are receiving information, may be getting a little smaller or fewer synapses. But then if you recover from the depression, you get increased size. And there's been actually quite a bit of work in animals and humans now looking at exercise, regular exercise, which has an antidepressant effect generally. And they see kind of similar. Did you do any measuring the volume, the size of different brain regions, or is it all functional?
we could theoretically with some tools we have. We haven't done it yet, no. But you bring out an important point. Let's go to this neuroplasticity, which has been shown for LSD, DMT, psilocybin, and even ketamine. So that's also this final path, that uses ketamine or a classical serotonergic psychedelic leads to this plasticity. And what we were surprised, we do animal studies also, that it's not just in the hippocampus. We were surprised that we found it in the frontal cortex also strongly. But we were disappointed when the dose were too high in mouse, used mice. Then we also had neuroplasticity in the amygdala, and that the mouse hasn't well responded to that. So it must be a dose range which is favorable to induce this neuroplasticity. The basic idea is that this neuroplasticity occurs, that the network opens up somehow in their connectivity, and then through the integration or through the post-acute experience, let's say training, you said learning, becomes very important. My understanding of learning on psychotherapy as we do it here is it doesn't help a lot. Let's say the network just opens up and then you hope for transformation to what? Angel, maybe. This is a little bit the idea of the humanistic psychology that people thought the higher the dose, the better you transform. But I don't think it's like that, because we have seen these high doses in the beginning that people were so anxious that you also can learn to become anxious, So it's a difficult thing, not a too high dose to keep inside in the process. And this learning must be important, the psychotherapeutic support or even intervention, not just being empathic to the patient after the session. Each psychedelic trip is followed in most of the current studies, at least 2 to 3 weeks follow-up with integration sessions where they speak about experience, the bad experience, the good experience, and how they deal with that. I think my understanding, what I see from our animal, what we see in the hippocampus with neuroplasticity and psilocybin, it's exactly that contextual learning is enhanced. And not just learning, in the context of something. That's really interesting. We none of the current study on this complex has looked what is the role or the meaning of the post-acute integration sessions. What do they really do? How important are they? If you skip them, what happens then? But because people don't want to go back to the 60s, they want to be sure people are safe.
Sure,
everybody's doing this integration.
So that brings us to the question of the use of psychedelics in popular culture, which is becoming increasingly common. I think in a lot of places, and definitely in the United States, they've been decriminalized in a couple of places, Washington, D.C., and I think the state of Oregon, and probably that will continue. So what should people be cautious about to make sure their experience is safe? And do you have any advice?
Yeah, so my advice is always that people should be aware, first of all, of the dosing, that they know the dose, that they still are aware who they are, that they are embedded from people who may not trip, some supervision, some guards or people there assist if there are emotional problems. Then certainly that they can talk, that they know these people, we realize it with our subjects that they get, when they had experience, they are with us, but then a friend or a family member picks them up from the hospital. So they're embedded, they can talk about, they can call us overnight if something is needed, or the next day they can come by. You need this, how do I say that, this environment who is understandable and not negative. If people are always negative, then it can fire back on feeling guilty or whatsoever, becoming more anxious. I think if we look at the numbers in controlled studies, the dose is the most important, the preparation and the non-so-called pharmacological factors, your personality structure. You know, most of the studies in healthy volunteers, we have done maybe 800 volunteers or so the past years, is a little biased because we haven't taken subject from if they had a schizophrenic patient. So we fear that that could exacerbate something, could break out. So the numbers that go around are mostly from, let's say, normal people. So
and they're all, are they young, just young adults, or a range of ages?
Yeah, in Switzerland, to go into such a study, you need to be 18. Most of them we have used people 20 upwards, but we had also very elderly people, because it was funny when we did some study and announced them in local newspapers, so homepages, and so we got calls from people that were 80 and 70 and said, oh, since 50 years, I'm waiting to get into a study. And I said, okay. But we had a restriction up to 65 because of cardiovascular thing. What we know is that all psychedelics, LSD and psilocybin, increase blood pressure about 20, we saw in Switzerland, millimeter Hg.
Yeah.
So you have to know you have to control blood pressure. So
well, exercise increases blood pressure too.
Yeah, you're right. With psilocybin, it doesn't last too long. We never had used any had to use any medication to bring it down. Mostly it was very transiently, sometimes, yeah, even nothing. But just to say that is the only well-discussed risk factor in clinical studies that has to be controlled for. It's not so, it depends on the country and regulation, whether it's an exclusion criteria. But in real studies, once a day, all these depression studies that are made now have to open up the inclusion criteria to, we just had a conference on that, because the reality is not young people. The reality, people with depression are, midlife or whatsoever, all the lives, there are all different forms of depression. It's not one depression, it's such a mixed bag. And it needs a lot of sophisticated controlled studies where you are prepared.
I don't know if this is true or not, but I read somewhere, I think it was Wikipedia, that Albert Hofmann periodically took LSD throughout his life. Is that true? Do you know if that's true?
I have known him very close for 30 years. I know this story. What Albert told me is that sometimes when he was about 70, 75, he tried 25 milligrams of LSD to look whether this is mood enhancing or gives you a little back of your vividness, but he did it rarely. It's always a myth, you know.
Okay.
Yeah. No, it's not something regular, certainly not. But he was certainly very open for experimentations.
And he lived to be 102.
Yes, yeah. And was very, it was, I don't know whether you have seen a video or something when he was 100, or he was very energetic, so unbelievable. He gave a talk. I had organized a 3-day meeting in 2005, and he was 99. and he had spoken as a speaker in the conference, like a young guy. He was 99. But then he was very philosophical, and we were all brain scientists. So, oh, wow, with 99. Is it due to LSD?
Yeah,
You know, it's
amazing.
but his wife, Anita, she was also 96 when she died. So they were very robust.
They had probably had good genes, huh?
Yeah.
Yeah. Okay. We covered a lot. I had a few other things I was going to ask, but
Yeah, you can go
one
for
is
it.
there's a method called transcranial magnetic stimulation.
Yes.
And I don't read the literature on this, but I was just thinking, seems to me and this is a way you can either increase the activity or decrease the activity of neurons in kind of fairly large areas. Has anyone found some sort of, an area and stimulation or inhibition level that caused hallucinations?
So far, we use it.
Oh, okay.
And we work here with a very old friend of mine, Julia Tononi in Wisconsin, who is that consciousness researcher. He introduced me to that, and with Marcella Massimimi and other people in Milano. Yes, there's a lot to say. First, it was used to block areas. People who had voices, schizophrenia patients hear voices. They used a sequence of stimuli that blocked and the auditory cortex. And apparently it helps. I haven't gone through that studies. I heard from other people that in the visual cortex, you can induce a phosphine-like flickering and blinking with certain sequences. But real hallucination, I haven't heard.
Yeah.
We had used it in a study. We go out with the paper soon. There's quite a different other idea. When you use TMS, transmagnetic stimulation, you have a magnetic field where you activate or deactivate, as you say, a certain brain regions. We use placebo and psilocybin and wanted to know, can we use TMS to modulate the psychedelic experience? It's the first study worldwide who went into this combination of technologies, simultaneously recording EEG, and look how when you give such a pulse, then the network immediately in milliseconds interacts, and you get information about in the three-dimensional space how brain areas interact through to the pulse. And you see, we call that an effect, cause-effect power. The cause is the magnetic field, and you actually see how it spreads. And we are running after that. And the crazy idea I had, I thought crazy because I thought, what would happen if we will block the visual system just in one half of the brain, some parts? How is the psychedelic experience then modulated?
Oh.
So at the moment, we have finished the study where we stimulated and analyzing this data, but the first paper may be ready in 2-3 weeks. I don't want to speak about the results because I don't want to interfere with the guys who did all the work. But the other thing will be very interesting. Can you block some areas?
But intuitively, since these act on the serotonin 5-HT2A receptor, and that receptor is distributed in meaningful places, maybe higher in certain neurons or concentrated in certain neurons. You would expect that the TMS is much more non-specific, and just intuitively, I think it would be less likely, very hard to induce a hallucination like you get with the psychedelic.
I think so. It will be hard. It will use a lot of energy as in terms of magnetic power to really bring them out of phase, that they get a kind of activated, in the elliptic state, where neurons fire like hell in hippocampus or other temporal areas. I think one could do it, but I'm not sure whether one would be allowed or whether it's ethical.
Oh, yeah.
I have no idea. It's extremely regulated with TMS, how strong you are allowed to stimulate. When you stimulate the motor area, you bend your finger, that's where we look at the threshold, that the pulse, instead that you, by will, bend your finger, the TMS pulse in the motor cortex, in the finger area, we are quite precisely. Let's say we stimulate in an area of 2 by 2 millimeters, you activate your fingers.
Oh.
And you bend
Oh,
them.
I see what you're saying. Yeah, there's this word called homunculus.
Yeah, exactly.
And so they're
Yeah.
called upper motor neurons, or so you can figure out exactly where.
Yeah. We do it, we make a normal structural MRI. We do it MRI guided. So it's very automatic done. The pulse, you have to imagine like a laser beam. We can measure how deep it goes into the brain by highly sophisticated feedback information we gather during the first pulse. Okay, it's too strong, it's too deep, we want to be there. And in the MRI scan, you see where you are. So it's three-dimensional guided stimulation. It's much too complex for patients.
That looks very complicated. So
yeah.
You're simultaneously
Yeah, 2 years to set up
doing
the experiment.
you're simultaneously doing TMS and MRI.
No, MRI we use as a guide where we are. We do
okay, and then
simultaneously we do EEG.
Then you move the patient.
Yeah.
Yeah.
They have an EEG cap on that we record the brain waves, and
okay, I got it.
we see the communication of we use almost 2 years that it's really smoothly running. It was smoothly running.
It sounds, I retired from my position as a lab chief at NIH a few years ago, and so I'm not as busy as you, and I know you're very busy. So
at the moment I'm still too busy.
I appreciate you taking the time.
Thank you.
To do this, and I'll put this up on the YouTube channel, and then I'm going to get these on one of the probably the audio part on Spotify and Apple. So
what I wanted to add is I had completely forgotten, but you may know that I was discovered by Mark Gayer from the Hefter. Have you heard about the Hefter Institute in the US?
Oh, go ahead and talk about that.
In '94, when we had the first conference on our very first data at the CIMP in Washington, Mark Gayer, it was an animal pharmacologist working, the only one in the world with psychedelics at that time, was a member of the Hefter Institute. And Hefter, this were these guys like Dave Nichols, Mark Gayer, Dennis McKenna, Charlie Grob, and they founded that in '93, and I became a member in '94. So I was for years the only guy who did some human studies. But it's important for me to have all these connections to the US and these wonderful people there that had supported me, because when you are alone, and you need all the support. But then, in 1912 it changed, and the Americans started their own stuff. So.
And so the COVID must have set you back, right? The COVID.
The clinical study became slowed down, yes, but not the lab development stuff. Yeah.
And how's the funding in the EU now?
The EU, it's good. I was founded by the Swiss National Science Foundation, like NIMH, and many times then we got the first EU grant for a transnational study, psilocybin in alcohol abuse. It includes France, Italy, Germany, and Switzerland. So it's a very nice, at all levels, epigenomics, fMRI, imaging, animal studies in Germany. It becomes really interesting.
Now something else popped into my mind. There's a lot of companies now, startup companies, and some of them, their idea, which I think, I don't know, makes any sense to me, is to make chemically modify psilocybin or try to make better,
Yeah,
but I don't know if that rationale is very good. I mean,
some people think, yeah, I know I've been approached by many companies. Some companies hope to find a 2A agonist that induces no psychedelic experiences. So
that'll be interesting if they do, to see whether.
So the question is, there's a debate whether is the psychological experience, which I think is an evolutionary benefit that we have consciousness and has some impact on learning and motivation and all these things, is important. But there are people, they are so strong behavioral, they say, oh no, it's just the neurons that change their habits, not the mind. So if we would have a psychedelic that directly activates, let's say, neuroplasticity, overgoing the 2A somehow down in the stream of interactions, then we have something we can use on a broader base. The psychedelic treatment is always a lot of preparation, support, integration. But if you can give it just as a pill, that's the big hope.
Well, yeah, people try to make money, and
sure.
Psilocybin and LSD, they're off patent. I assume that, they've got to be off patent. They're off patent, so they may add a hydrogen to the atom, and it has the same. It has the same effect, and it's
I'm also surprised about this big hype, and I make sometimes jokes. There are more reviews from people who have never worked with these things than clinical studies.
Oh yeah, that's
hundreds of reviews now.
Go on PubMed, and there's a lot of review articles, but not.
And then you have 12 studies, 200 reviews. That's life.
Yeah. Okay, friends.
It was a pleasure. Thank you so much.
Yeah, great to meet you and talk to you. And I hope you have a good spring and summer.
You too. Bye-bye.
Okay, bye.
Thank you.
Yeah.